allergy session. And our first speaker is Jenny Hamilton, speaking about characterization of autoreactive B cells in BXD2 autoimmune mice using B cell tetramers. And Jenny works with John Mounds, UAB. For those speakers, we're going to do two things in order to keep people on time. I think you'll be able to see there the 15 minutes that you had. I also have a timer here on my cell phone. At the end of 11 minutes, you'll hear kind of a little crunch, and then we'll set the timer for a minute, and you're going to hear a plane chime, okay? So then you put your seatbelts on and get back in your seat. So you'll have 3 minutes after that for questions, okay? So.
Okay, good afternoon, everybody. I'm happy to be here today to share with you some of our work using B cell tetramers in BXD2 autoimmune mice. So as you all know, B cells are critically important in autoimmune disease, especially in SLE where B cell tolerance is so profoundly affected. So a major challenge is to isolate these autoreactive B cells from autoimmune subjects so that we can study them. So the major objective of the present work is to use B cell tetramers to isolate autoantigen-specific autoreactive B cells. In our lab, we study the BXD2 mouse, which spontaneously develops a progressive autoimmune disease characterized by very high autoantibody titers. And you can see here the anti-DNA autoantibodies, anti-DNA IgG. So we're very interested in these autoantibodies because from hybridoma studies we know that these antibodies can be very pathogenic and can transfer disease. So previously we've characterized the autoantigen targets of these antibodies in BD2. You see here using a 2D gel analysis and time of flight mass spec, we've previously identified the autoantigens that BD2 targets. And among these are several mitochondrial enzymes, also several proteins from the heat shock family, and also, importantly, several histone proteins and also several of the ribonuclear proteins. We've also characterized the cellular abnormalities in the BD2 spleen, and we've identified that the marginal zone precursor B cell population is expanded in BD2. And we can see that here by FACS, we see the expanded MZP population compared to B6. And the lab's published several papers on this marginal zone precursor B cell, but just briefly, this cell is a very co-stimulatory cell and a very potent deliverer of antigen from the marginal zone into the germinal center. And we can see here, it probably would be better if the lights were off, but here these cells in yellow are the marginal zone precursor B cells, and they don't localize to they don't stay in the marginal zone. They actually can transport antigen into the germinal center light zone, localized with T cells. So one of the first things we wanted to do with B cell tetramers is to determine if these marginal zone precursor B cells are specific for autoantigen. So this is a summary of our experimental strategy to identify BD2 tetramer positive autoreactive B cells. The first step is we identify autoepitopes, and we do this using a peptide microarray, and I'll talk more about this in just a second. The second step is we do an epitope analysis, looking at the 3D structure, 3D crystal structure of the proteins, and also using Discotope, which is an epitope prediction program. Once we select the top peptides, we synthesize them, and we do ELISA screening with our mice in the lab. And from there, we pick the very best peptides, and we generate tetramers that can be used then to isolate antigen-specific B cells in our mice. So this is step one, where we identify the autoepitope repertoire of BD2. We use this autoimmunity peptide microarray. So you can see here, all of these red dots correspond to linear peptide autoepitopes from the immune epitope database. So we probed this chip with BD2 serum, and we identified a lot of positive hits. Thankfully, the peptides identified by the array matched those that were previously identified by the 2D gel I showed you before. So the U1 SNRP, several epitopes from this antigen were high, also several epitopes from the HSP60, the heat shock protein family, as well as some others. So the advantage here is that now we actually know the peptide epitope, whereas before we just knew the antigen. So this is something that we can use to make a tetramer now. So as you can see from the previous slide, there were a lot of peptides on the chip, about 3,000. So we narrowed these down to 200 or 300 based on the intensity of the score. And from there, we had to narrow it down further. So we did that using, looking at the 3D crystal structure and also using a Discotope, which is an epitope prediction program. And so, for example, here we see the 3D crystal structure of the U1 SNRP, and this is one of the epitopes that was identified by the peptide microarray. And we can look at the crystal structure and see that this epitope, it looks like a nice linear coiled coil epitope that would be highly exposed and a good linear epitope for a B cell. So based on this, we selected the top peptides, we had them synthesized, and then we went back to our mice and we screened mouse serum with our peptides using a neutravidin-based ELISA. So in this assay, BD2 reacted most strongly with these nuclear epitopes. These are peptides from, all of these are peptides from from RNA binding proteins. So SMD1, lupus law, and the SNRMPs were very high in BD2 while they were low in B6. We're also interested in citrulline modification in autoimmune epitopes. So we also selected a fibromodulin, a citrulinated fibromodulin epitope to generate tetramers from. We can also use these peptides in ELISA spot. So see here the different peptides here that we coat the plates with. And we see that it doesn't really matter the stimulation condition, IL-21 or PMA, inomycin. In all cases, BD2 does make B cells that produce autoantibodies that react with these peptides, while B6 does not. So at this point, we're ready to make tetramers, and we've adapted the method that's established by previous investigators, where we use streptavidin here, conjugated to biotinylated peptides. And all of this is labeled with PE so that we can detect and isolate these cells. We also use a non-specific tetramer that controls for all of the B cells that stick non-specifically to the tetramer. And this is labeled with PE conjugated to AF647 so that it runs in a different channel. And we can plot 2D, we can make a 2D plot here, and we see the non-specific tetramer on the y-axis and the specific tetramer on the x-axis. And we see the single PE positive B cells here. These are the tetramer positive population that we're interested in. And one thing I forgot to say is we do a anti-PE magnetic bead-based enrichment process to enrich for these cells. So looking at some of our early experiments testing these tetramers in our mice, we see we used fibromodulin, the citrulinated fibromodulin, SNRMP, and SMD1. And in the first case with fibromodulin, we see a marked expansion of tetramer-positive B cells in BXD2 compared to B6. We also see an expansion in the SNRP positive population as well as SMD1, but to a lesser extent. So we knew that it would be important, since there's not great frequency differences here between B6 and BD2, we knew it would be important to look at the phenotype differences in tetramer-positive B cells between B6 and BXD2. And the first thing we looked at was the CD21, CD23 plot. And looking at the three tetramers, LA, SNRP, and SMD1, we see that all of the B cell subsets are represented here, but one consistency that we see in each case is that the marginal zone precursor B cell is expanded in BD2 as compared to B6. So we wonder what might be stimulating these MZP tetramer-positive cells in BD2 mice. So because we've previously identified that BD2 MZB marginal zone precursor B cells upregulate CD69 and CD86 in a type 1 interferon dependent manner, we look at these markers within the MZP tetramer-positive population, and we see that these cells that are upregulating, these cells upregulate CD86 and CD69. So this shows that these MZP B cells that we know so much about and we know are a problem are also autoantigen reactive. So in summary, we've demonstrated a system here whereby we can identify linear peptide autoepitopes that we can then use to, we've demonstrated a system where we can identify linear autoepitope peptides that can then be used to generate B cell tetramers that can be used to generate tetramers that can be used to isolate these B cells in an autoimmune subject. We further identified that the MZP population is enriched in autoantigen specific B cells, and that they further upregulate CD69 and 86. In the future, in the immediate future, we want to investigate these tetramer-positive cells in interferon alpha receptor knockout BD2 to characterize how these tetramer-positive cells interact with T cells at the pre-germinal center, germinal center, and post-germinal center stages that finally lead to the high titers of autoantibodies. So last, I just want to thank the wonderful members of the Mounts Lab at UAB who have all contributed a great deal to the work presented here.